Kira and Sabrina's AIDS Project.
The human immunodeficiency virus, or HIV, is a type of retrovirus. Retroviruses are viruses that contain a single RNA strand. HIV can infect a number of different cells and particularly targets white blood cells such as CD4-bearing macrophages and T helper lymphocytes. HIV attaches to the CD4 molecule on the cell surface via envelope GP120 on the HIV. The envelope protein assists attachment of the virus to the cell surface. A conformational change occurs in GP120, allowing it to bind to the chemokine coreceptor. In the absence of CD4 binding, ENV cannot interact with the coreceptor and infection does not occur. The HIV envelope fuses with the cell membrane via a structural change in the GP41 envelope protein. The nucleocapsid containing the RNA genome enters the cytoplasm of the cell. The outer layer of the capsid is dissolved as it enters the cell, leaving the proteins inside the capsid. Reverse transcriptase takes the viral RNA and, using host nucleotides, converts that viral RNA into a single strand of DNA. While it does that, it makes some random errors because it has very poor proofreading activity. That single-stranded DNA is again reverse transcribed into a double-stranded DNA. An enzyme called integrase grabs hold of that double-stranded DNA and carries it through a nuclear pore into the nucleus of the cell. Within the nucleus of the cell, it finds the host chromosome and makes a nick in the host DNA and allows for HIV to insert itself into the host chromosome, establishing a lifelong connection. From that DNA, the RNA polymerase makes messenger RNA. This mRNA uses the new genetic information to transcribe for certain proteins needed to create an HIV molecule. These strands of mRNA move out of the nuclear envelope into the cytoplasm and translate the genetic material onto ribosomal RNA. These different strands of mRNA create envelope proteins and other viral proteins which will be used to make up the key components that the virus will need. All of these different proteins make their way to the surface of the cell. This then buds off at the cell's surface. This is an immature virion because the polyprotein chain needs to be digested into its component parts to become mature. This is done by an enzyme called protease. Protease breaks up the polyprotein chains and ultimately allows for them to coalesce and form the mature structures that make the final virion. This is a mature infectious virion.